Dear students, today I am going to discuss about the structure of different types of immunoglobulins, because the structures of immunoglobulin I have already discussed in my last lecture. So today I am going to discuss about the types of immunoglobulin, different types of immunoglobulins and their functions. Then I will cover immunoglobulin genes and diversity of antibody. So first I will start with immunoglobulin G. If you look into the figure, then you can see that immunoglobulin G or IgG, they are in their monomeric form. And this particular monomers of immunoglobulin G are most versatile immunoglobulin because it is capable of carrying out all of the functions of immunoglobulin molecule mostly. And in serum, 75% of the immunoglobulins are IgG, and maximum daily production of IgG among the all other type of immunoglobulins, and their self life or the half life is the longest one. It is of 23 days. IgG have four different subclasses: IgG 1, 2, 3, and 4, and all are different. The difference there in their biological functions, in their uh, chain length of amino acid length of each region, as well as the number of disulfide bridge bonds. Now I'm coming to the function part of IgG. So IgG is an antibody that can cross placenta and provide immunity to fetus and newborn. They can also take part in complement fixation process. The Fc region of the IgG can bind to the complement factors and activate the classical pathway of the complement system. They can also induce phagocytosis. Here, the two subclasses, IgG1 and IgG3, they bind to Fc receptors on phagocytes with high affinity and enhances the phagocytosis of antigen bound to it. The other functions are the mediate precipitation as well as neutralization reaction, and the neutralizing major role they play is the neutralizing the toxins. Next one is IgM. Here, this IgM is a pentameric form of immunoglobulin. Normally, they exist as a pentamer, but they can exist in monomer. And in the pentameric form, all the heavy chains and the light chains are identical. You can see all the light chains and the heavy chains are identical, means all the monomers are identical. This particular immunoglobulin has an extra domain on the main chain. This is the extra domain. Here you can see this is the extra domain of the here, and it has another protein covalently bound via disulfide bridge bond called the joining chain of the J chain. This one is the joining chain of the J chain. This chain functions in polymerization of the molecule into the pentamer. So they are only joining the monomers of IgM and are responsible for its pentameric form. And due to having this pentameric form, their molecular weight is the largest one. So highest molecular weight of among the all other types of immunoglobulins, and that is why they are called as macroglobulin. So like IgG, IgM also function in complement fixation, in agglutination, in toxin neutralization, and the other function they are performing is opsonization. So they are actually coating a particle with proteins and facilitating phagocytosis process of the particle by tissue macrophages and activated follicular dendritic cells, as well as binding the receptor on the PBMC's peripheral blood cells. Next one is IgA. It's a dimeric form of immunoglobulin. And it contains two subclasses, IgA1 and IgA2. So IgA1 contains alpha 1 heavy chain, and this particular immunoglobulin subclass of IgA is found in serum, and most lymphoid tissues have a predominance in IgA1 producing cells. Whereas IgA2 have different alpha 2 heavy chains, and there the heavy and the light chains are not linked with disulfide bridge bond. There's no covalent bonding is there. This is a difference among the other immunoglobulin with the other immunoglobulins, because there is no covalent bond that is attaching the light chain and the heavy chains. But instead of this covalent bond, there are some other non-covalent interactions like hydrophobic interactions, hydrogen bonding, ion dipole interactions, all which is. So in secretory lymphoid tissues like the gut-associated lymphoid tissues or GERD, so they are predominant. And the share of IgA2 production is larger than in the non-secretory lymphoid organs. So secretory IgA, they are also dimeric in nature, and two monomers are joined by this J chain or the joining chain. And they are located mostly found in milk, saliva tissue, saliva tears, etc. And their functions is local as well as mucosal immunity. Mostly they function against, they're very much effective against few bacterial species like Salmonella dupi or Neisseria, because they are, in case of breastfeeding infant, they are resting at the infant gut, and also responsible for protection against some gut pathogen. That's specific pathogen. Next one is IgE. So IgE production in serum in human body is the lowest among the other type of immunoglobulins, and also it has shortest half-life, minimum daily production, and also lowest concentration of serum. So mostly they have the affinity to the for the surface of tissue cells or the mast cells, and their functions is that they are actually functioning as a potent mediator of type 1 hypersensitivity. And it has been noticed that in case of helminthic infection, their production is elevated. The last one is IgD, is found in membrane IgG on the surface of the B lymphocytes, and actually they are functioning as a B cell receptor along with the IgM. This is the function of IgD. Now I'm coming to antigenic determinants of IgG immunoglobulins. Since antibodies are glycoproteins, they can themselves function as a potent inhibitor. And also they can induce antibody responses in the host. So likewise the antigen, the antibodies also, the immunoglobulin molecules also, no entire immunoglobulin molecule is immunogenic. Only a part of the portion that is known as the antigenic determinant, the specific site which is known as a epitope. This is actually responsible for immunogenicity, not the whole immunoglobulin molecule. Based on the location of the antigen determinants, immunoglobulin molecules are divided into three different types. They're called isotype, idiotype, and allotype. What is isotype? So it's an unique amino acid sequence that is common to all immunoglobulin molecules of a given class of species. So far we have 5 different immunoglobulins, and their subclasses are also called isotypes. So they vary from each other in amino acid sequence of constant regions of their heavy chain. Here you can see this is the isotypic determinants, means here they vary from each other in amino acid sequence of the constant region of the heavy chain. The next one is idiotype. So again, the unique amino acid sequence present in the paratope region. This is the antigen binding site, the paratope region, in the variable heavy chain as well as variable light chain region. of one member of species. It is found in the amino terminal end. So this is the amino terminal end that is closer to the antigen binding site. So the amino terminal end of both the light chain as well as the heavy chain. And they function together. They serve as antigen binding site as well as antigen recognition site. Next one is the allotype. So this is antigenic specificities which distinguishes immunoglobulin of some class between groups of individuals in the same species are called allotypic specificities. So next one is the immunoglobulin genes. So antibodies are proteins, and proteins are evolved by or encoded by gene. Antibody diversity therefore poses a special genetic problem. So how can an animal make more antibody than there are genes into its gene? This problem is not quite as formidable as it might first appear. So if you recall that variable regions of both the light and heavy chains of antibody usually form the antigen binding site. Thus, an animal with 1,000 genes encoding light chains and 1,000 genes encoding heavy chains could, in principle, contribute their product into 10 to the power 6 different ways to make 10 to the power 6 different antigen binding sites. Nonetheless, the mammalian immune system has evolved unique genetic mechanisms that enable it to generate an almost unlimited number of different light chain and heavy chains in a remarkably economical way by joining separate gene segments together before they are transcribed. So here we are going to discuss about the mechanism that the B cells use to produce antibodies with an enormous diversity of antigen binding sites. We will then consider how a B cell can alter the tail region or the stem region, means the constant region of the antibody it makes while keeping the antigen binding sites unchanged. This ability allows the B cell to switch from making a membrane-bound antibody to making secreted antibodies, or from making the class of antibody to making another, all without changing the antigen specificity of the antibody. So here you can see this picture here, a mouse embryo cell that is not making an immunoglobulin, and here the mouse B cell that is specific for making a specific light chain. And when DNA is extracted from both the chains and digested with restriction in the endonucleases, and then the DNA restriction fragments separated by electrophoresis, and both the variable and the constant regions coding sequences visualized by hybridization with radioactive DNA probe. Then we have found that three different regions, three different fragments. One is C region coding sequence, V region coding sequence, and the third one is a V and C region coding sequences are on same fragment. So the first direct evidence about the immunoglobulin genes, that DNA rearranged during B cell development, came in 1970 from the experiment in which one of the molecular biologists compared DNA from here the mouse embryo, and it do not make antibody with the DNA of a mouse B cell tumor here, which makes a single species of antibody molecule. from here, the single species of antibody molecule. The specific variable region and the constant region coding sequences that the tumor cell here, the tumor cell B cell use are present on the same DNA restriction fragment here, same DNA restriction fragment in the tumor cells, but on two different restriction fragments here in the embryos. These rearranged at some stage in the B cell development. And this is very important for diversity. So we know that each type of antibody chain, kappa and lambda light chains and the heavy chains, has a separate pool of gene segments and exons from which a single polypeptide chain is eventually synthesized. And each pool is on a different chromosome and contains a larger number of gene segments encoding the variable region of an antibody chain and a smaller number of exons encoding the constant region. During the development of the B lymphocytes, a complete coding sequence for each of the two antibody chains to be synthesized is assembled by a site-specific combination. In addition to bringing together the separate gene segments and the C region exons of the antibody gene, these rearrangements also activate transcription forms of the gene promoters. So changes in the relative positions of the enhancers and the silencer acting on the promoter. Thus, a complete antibody chain can be synthesized only after the DNA has been rearranged, as we shall see the process of joining gene segments contributes to the diversity of antigen binding sites in several ways. So here you can see that each variable region is encoded by more than one DNA segment. So when the genomic DNA sequences encoding both the variable and constant regions were first analyzed, it was found that a single region of the DNA encodes the C region of an antibody chain, but two or more regions of the DNA have to be assembled to encode each variable regions. And each light chain of the variable region is encoded by a DNA sequence assembled from two gene segments, that is the long V gene segment and the short joining, that is known as the J gene segment, which is not at all similar with the J chain of immunoglobulin, which is encoded elsewhere in the genome. So the figure here will illustrate you how this is happening. Means genetic mechanism involved in producing the human kappa light chain polypeptide from the C region exons and the separate G and G segments. So this is a germline DNA isolated from human, and here you can see these are the regions of DNA to be joined. So DNA rearrangement is taking place during the maturation or development of these cells. So from the germline DNA, what we are getting the B cell DNA. So one is your B cell, one is your germ cell. So in this figure, what we are getting here, this is your germ cell, embryo cell, this is your B cell. So here the DNA sequence is very large, here the DNA sequence is not such. So now you can see that from the germline DNA, now we are getting the B cell DNA. And here you can see some parts, some portion, this is the region to be joined, now they are joining. So this region is sized out from P40, J1, J2. So this section is excised or removed, and this portion is joined with B1, B2, and B3. And it's not that they are going for transcription directly. Then the RNA transcript that is coming from the B cell DNA via transcription, there you can see they are again removing V1 and V2, and then conserving B3, J3, J4, and J5 and C. So C is responsible region, that is the sequence that is responsible for encoding the C region of an immunoglobulin. And these are the sequences that are responsible for the variable kappa as well as the lambda sheaves. So again we can notice RNA splicing removing the introns, and then this mature mRNA will directly go for translation or undergo translation to produce the light chain as well as the heavy chain. So here you can see that only we are focusing on the light chain, not the heavy chain. And here in the light chain, there are two different regions. One is the variable region, one is the constant region. And C is the gene that is encoding the constant region, and the two other genes, B3 and J3, they are actually encoding the variable region. This is known as VJ joining. So the VJ joining process involved in making the human kappa light chain. So here each heavy chain has a V region, is encoded by a DNA sequence assembled from the three gene segments, that is the V segments, the J segments, and the diversity segment, that is known as a D gene segment. And this figure will show you the number and organization of the gene segments used in making human heavy chains. So here you can see this is your germline DNA. This is not at all the V cell DNA. This is the germline DNA. So there are three different segments. One is the V segments, variable region. One is the J segments, joining segments, and the diversity segments. So V, D, and V, J, and D. So this is the variable segment. This is the diversity segments, and this is the joining segment. And apart from that, you have this constant segment. So there are 51 V segment, 27 D segment, 6 G segments, and an ordered cluster of C region exons. This is the cluster of C region exons: alpha, beta, gamma, delta, epsilon, and mu. So this encoding a different class of heavy chain here. It's not a light chain. First one was the light chain, and this one is a heavy chain. The D segment and a part of the J segment encodes amino acids in the third hypervariable region, which is the most variable part of the region. And this figure is not drawn to scale. You can see here it is not drawn to scale. And the total length of the heavy chain locus is over 2 megabases. Moreover, many details are omitted. So for instance, each C region is encoded by multiple exons. Here you can see the exons. There are 4 clusters of C gamma regions, so gamma 1, 2, 3, and 4. And the variable heavy chain gene segments are clustered on the chromosome in groups of homologous families. So the genetic mechanism involved in producing the heavy chain are the same as those shown in this figure for light chains, except that 2 DNA rearrangement steps are required instead of 1. First is a D segment joins to the J segment, and then the V segment joins to the rearranged DJ segment. So we are getting 2 regions, 2 segments. One is the DJ and next one is VDJ. Now what is antibody diversity? So we know that there are millions and millions of antigens. So they have millions of epitopes. Our immune system has the ability to produce a specific antibody, means a variable region against all antigens. And this diversification of antibody production is actually known as antibody diversity. Now, to understand the diversification mechanism in antibody generation, we must know the genetics in immunology regarding these 3 different facts. One is the multi-gene organization of the IgG genes. Then the variable region gene rearrangement. So first one is the VJ and then VDJ recombination that we have already discussed in our previous slide. And the third one is the mechanism of recombination. So here generation of antibody diversity. So 7 means to date of antibody diversifications have been identified in human. And these 7 means are multiple germline genes, combinatorial VDJ joining, P region nucleotide addition, N region nucleotide addition, junctional flexibility, combinatorial association of the heavy and light chains, and then somatic hypermutation. So the first one is the multiple germline segment. What is that? So there the kappa chain and the lambda chain of the light chain and the heavy chain are coded by separate multigene families situated on different chromosomes, not on same chromosome, different chromosomes. And since these immunoglobulins are the product of multigene family, different kind of immunoglobulins are produced from different genes. So here, for an example, you can see human IgG gene pool, the chromosomes here in human and mouse. If you compare, then you can see the genes that is for the lambda chain for human is 22 and mouse is 16. For kappa light chain, human is 2 and mouse is 6, and for heavy chain, human is 14 and mouse is 12. This is statistics. That, not the statistics, this is the location of the gene. So combinatorial variable, then joining light chain, and then variable, and then VDJ joining and heavy chain that we have discussed. So here we need to remember a few things to recognize this recombination. So first thing is that the immunoglobulin have 2 chains, heavy and light chains. They have 2 regions, variable and constant region. The variable region of the light chain is coded by one of the VJ recombinant, and the variable region of the heavy chain is coded by one of the VDJ recombinant. So 2 recombinants, VJ then VDJ. So the C region of both H and L chains is coded by one of the C segment. So there are multiple genes in C. So for an example, here you can see the lambda chain gene family. They have 31 B lambda, 4 J lambda, and 7 C lambda segments. And this is an example of a mouse lambda chain. In case of kappa chain of a human being and immunoglobulin kappa chain gene, so 40 VK, 5 JK, and single CK. But if you go for the heavy chain gene family, then you can see the variable region is coded by 3 type of gene segments, that is V, D, and J. And here you can see the constant region is coded by the C region gene, and the C region gene is in the order of C mu, lambda, C mu, delta, gamma 3, gamma 1, gamma 2b, gamma 2a, C epsilon, and C. The constant region of the C region gene is coded by 51 VH, 27 VH, and 6 VH genes. So here is the arrangement of the heavy chain of DNA. So if we want to sum up, we can say that the immune system is capable of producing a tremendous diversity of antibody specificities, and the IgG genes are assembled during the B cell differentiation from different gene segments that are combined through the site-specific recombination. The variable regions are encoded by the variable diversity and joining segments, and the human immunoglobulin heavy chains locus contains approximately 50 VH, 20 DH, and 60 H immunoglobulin gene segments. The IgG of a kappa locus contains 100 VK5JK, whereas the IgG of lambda contains 30 V lambda and 8 J lambda. So these different segments in the H chain locus are joined by the recombination as outlined in the figure mentioned there. And the L chain locus undergoes a similar recombination process, with the exception that it lacks the D region genes. Further diversity is generated through junctional diversification. That is, nucleotide can either be lost or added. Nucleotides are P and N nucleotides in the joining of VDJ segments in case of heavy chain. So additional diversity can be derived from pairing of the V, L, and VH regions. So furthermore, after the pre-lymphocytes has been stimulated by antigen, a somatic hypermutation mechanism may operate on the assembled permeable regions.